Hello, I'm Jeanette, and my other team members are Nicole and Jan, and this is exploring the effects of temperature on C. californica through time.
So the species studied was the Cerathidiopsis californica, also more commonly known as the California horn snail. They often reside in salt marsh-dominated estuaries of the upper coastal intertidal zone of California. They have terraform-shaped shells growing to adulthood at roughly 25 millimeters in length, or about 1 inch. The horn snail grow and reproduce from spring to fall with the average lifespan of 6 to 10 years, which applies to both uninfected and infected snails. They often feed on microalgae or benthic diatoms as a source of food within their coastal ecosystem. A little more about horn snails in their environment. A study in 2010 stated that the parasites that often reside in these horn snails are more resilient to the increase in pH and the salinity levels in water. This goes hand in hand with another study in 1993 that suggests that the horn snails had adapted to mature at a smaller size in order to prevent the risk of parasitic castration. Additionally, a study from 2012 mentioned that the shades created by plants in the salt marshes reduces the density of benthic diatoms, which is the main food source for these horn snails, which then may reduce the population density overall. Marine species are being negatively impacted by climate change, which is quickly becoming a more prevalent issue year after year. With the rise in human activities, the greenhouse gas emissions has been increasing at a significant amount, which in turn causing a rise in the overall temperature, especially in the habitats of the C. californica. Ocean acidification would occur due to the higher concentration of carbon dioxide in the atmosphere entering the water. And additionally, the salinity in the ocean would increase as well due to the higher rate in evaporation with the increase in temperature of the climate. The purpose of the study is to look at the effects of changes in climate through the span of 4 years, from 2019 to 2022, on the shell size of the Cerathidiopsis californica. And here we have a general graph of the global land and ocean temperature. It shows an incline from 1880 to now.
So, with this knowledge about the horned snails, our main discussion question was, how does temperature actually affect these snails in the Newport Aquatic Center? So based on this question, we have our null hypothesis of horn snails' shell sizes decrease as their environment temperature increases due to climate change affecting calcification, and our null hypothesis being that there would be no shell size differences in the snails based on temperature. So to help answer the question in mind, we did our research at the Newport Aquatic Center, and our research design consisted of having 5 groups that were made. These groups were assigned either shoreside or seaside locations at random. We did that by flipping a coin. So within these groups, we assigned roles like data recorders, counters, and measurers. So after everyone received their roles in their groups, two 30-meter transit tapes were placed along the shore, and the data collected based on our assigned locations was size in millimeters and population count. So once everybody was ready, it was time to do our data collection. And we did this by setting down quadrants along the transit lines based on our assigned locations. Then the measurer would record the found snails with a caliper, and 10 snails were measured per location. And baskets would be used to collect near muddier areas to prevent anyone sinking into the mud. And the recorder during this time would be writing down the data given by the other team members. A water collection was also done by using a refractometer used to collect dissolved oxygen levels, salinity, and density levels.
So now I'm going to be taking you through some of our results. So our first graph here is figure 1 that is showing the relationship between temperature and average shell size for each of our sampling days. So on our x-axis, we have all of our sampling dates. On the left y-axis, we have temperature in degrees Fahrenheit, and that's represented by the box and whisker plot. So if you take a look at the box and whisker plot, where the orange meets the gray, that is the average temperature for that day. And the top and bottom whiskers show the variability of the temperature between 12 p.m. and 6 p.m., which is when the samples were obtained. And then on the right y-axis, we have average shell size in millimeters for each of the sampling days. All of the individuals that were measured on that day, we took the average of those shell sizes. So based off of our hypothesis, we should be seeing some sort of relationship between temperature and average shell size. So we believe that with a lower temperature, we expect to see larger shell sizes, and with a higher temperature, we expect to see lower shell sizes. Looking at this graph, we can kind of see this relationship happening. So on January 29th and September 2nd, those are our lowest temperature days. And we do see that the shell size observed during those days is the greatest out of all the other sampling dates, but we're not entirely sure if there is a statistical difference between these relationships. So I went ahead and ran a regression test because we have two quantitative values. And with the regression test, we got a p-value greater than 0.05.
Here we have figure 2a, which shows the C. californica size distribution from 2019, with the top graph being from the seaside transect and the bottom being from the shoreside transect. And this can also apply to all of the other size distribution graphs as well. We have the horn snail count on the y-axis while we keep the size range on the x-axis. And as you can see, on the 2019 data, we have a p-value greater than 0.05, with the range being roughly in the 19.1 millimeters to 25 millimeters. As for the data from 2020, we have a range from 21.1 millimeters to 24.9 millimeters, with a greater p-value as well. Then we have figure 2c and 2d, which respectively shows the data from 2021 and 2022. Both of these data have a p-value below 0.05, with ranges roughly from 19 to 30 millimeters.
So there's some information that we've already gathered from primary literature. So we know that C. californicus is a species that is known to survive in hotter temperatures. And we also know from the literature that shell size is expected to decrease with decreasing pH levels. So we know from our ocean acidification section, when we have a decrease in pH, there is going to be more bicarbonate ions produced instead of carbonate ions, which are what these invertebrate shell-producing species need. They need those carbonate ions. So we're expecting to see the shell size to decrease with decreased pH levels. However, based off of our results, we found that there is no significant difference between the size of individuals spotted on seaside and shoreside transects in 2019 and 2020. So our p-value was greater than 0.05. And we also saw no significant difference between temperature and its effects on shell size across all of our sampling dates. And we got a p-value greater than 0.05. However, we did notice that there is a significant difference between the size of individuals spotted on the seaside and the shoreside transects in 2021 and 2022. So we got a p-value less than 0.05. So some future implications that our data has, we all know that our carbon emissions are expected to increase, and the increase of carbon emissions are going to have an increase in temperature. So this graph here was sourced from climate.gov on the right, and it shows the expected increase of temperature up to 2100. So there are three different scenarios here, but basically what you need to understand about this graph is that no matter what scenario we pick, temperature is going to be increasing in the future. And we know that an increase in temperature leads to an increase in primary productivity. And an increase in primary productivity actually causes an increase of food supply for C. californica. However, right now, we know that the climate is constantly fluctuating. So we're currently in La Nina conditions. So we expect cooler temperatures and less productivity because of this. We also know that C. californica ingests relatively small diatom sizes. Diatoms are their main food of choice. And Whitlatch et al. Believes that this is likely related to the length of the snail. And so we will expect to see a decrease in diatom biodiversity. And if we notice a decrease in shell size in the horned snails in coming years, because temperature and CO2 is increasing as well, then the smaller the snail, the more likely they're going to switch to a smaller food source. And this could potentially cause competition with other species as well. So it's really important that we understand how temperature is going to affect the shell size of these animals.
Some errors in the study for historical data, groups did not measure the same number of sample sites each year, which can skew the results. And also, no other data besides count and size was recorded when performing the lab. These are our references. And finally, we just want to thank the other members of Group 5 that helped complete this lab, Chelsea, Dr. Tran, Newport Aquatic Center and the Snails, and the past classes of marine ecology. Thank you.